I hope my slides are visible.
Yes, ma'am.
Yeah. Welcome to host Pathogen Interactions Immunology from NPTEL courses. This is week 2 live session. I am Sumati Rafiraj, PMRF scholar from University of Hyderabad, and the course instructor for this course is Professor Himanshu Kumar from ISA. So, as we know, the course is from 20th of Jan till 3rd Feb, where the enrollment ends, and the exam date is 4th May, and exam registrations are currently going on, and it ends till 14th. Okay. So last week we discussed about week 1, and the other somebody's mic is on. Can you please mute it? Yeah, thank you. So last week we discussed the first week's assignment indirectly in few topics. We will do the same this way, and I had also informed that if there were any queries or if you want any topic in particular, you can mail me, but I do not receive any mail. So I will assume that we will go on with the same mode with the live session. So in week 2, the topics that were covered were immune organs 1 to 3, cells of immune system, hematopoiesis, and then transgenesis 1 and 2. So depending on the week's assignment, I have briefly discussed few topics which will help you in answering every question there. So other than this, if you want me to discuss anything or if you have any doubts, we can either discuss it at the end of the session if we have time, or you can mail me like I said last week to the mail ID that is given here, 20lbph04@uhyd.ac.in. So that I can, if you want me to discuss something else, I can do it in the next week's session. Okay, so coming to the topics, first we will know a little bit about the lymphatic system. So the lymphatic system includes lymph nodes, lymph vessels, bone marrow, spleen, thymus, tonsils, adenoids. So your lymph tissues are also the parts which are in your small intestine that also comes under your lymph tissues. So lymph vessels are thin tubes that carry lymph and white blood cells. So in this diagram here, you can see representation of all these organs that are part of your lymphatic system. So your adenoids are present here, and your tonsils are present here near your throat, and the mucosa associated lymphoid tissue in your nasal cavity that also comes under your lymphatic system. Then thymus is one of your major lymphatic organs, and then you have your spleen, which is located here. One second. Yeah. Then you have your spleen, which is located here, and then the lymph tissue in your intestines. Then your bone marrow, that is also your lymph comes under your lymphatic system, and your lymphatic vessels, which carry your lymph. And in this system itself, in your lymphatic vessels, they all diverge at your lymph nodes. Okay, so these are the major uh, parts of the lymphatic system that we have to keep in mind. And the lymphatic system is important for the body's immune system because they carry your white blood cells, your lymphocytes, and then it helps in fighting your pathogens, your antigen excretion, etc. So, a blockage in the lymphatic system is called lymphatic obstruction. It can cause lymphedema, which is a chronic swelling in the body's soft tissue. So, as you can see, your lymph here is passing through your lymph. Someone's mic is on again. Can you please mute your mics?
Sorry, ma'am. Sorry.
Yeah, please. Okay, so as you can see, your lymph passes through your lymph vessels, like your blood vessels. So if there is any blockage in these vessels, it will obstruct the flow of your lymph, obviously. So that is your lymphatic obstruction. And once this obstruction is formed, then your free flowing lymph will get accumulated before the obstruction or after the obstruction, and uh, it keeps on accumulating, and it will eventually lead to swelling, and there will be retention of lymph in that particular area. So this swelling due to your lymph accumulation is called as lymphedema. Edema is nothing but swelling. That swelling which is due to the accumulation of lymph is called as lymphedema. So this lymphedema can be very serious because it is involved in your immune system. So if there is no free flow of lymph, your immune regulation also will be disrupted, and this can lead to several chronic conditions. So if there is any such thing, you should immediately consult a physician. So your lymphatic system infections can affect the lymph nodes, lymph vessels, and lymphatic system as a whole. So any part of your lymphatic system is uh, probably exposed, or probably affected by infections once your pathogenic bacteria, virus, or fungus enters your body. And these infections can be caused by bacteria, virus, or fungi. Types of lymphatic system infections. So there are several types. So the basics are lymphadenitis, an infection of one or more lymph nodes, often caused by bacterial, viral, or fungal infection. Symptoms include enlarged, painful, or pus-filled lymph nodes. So as we discussed, your lymph vessels here, they come and converge at your lymph nodes, you can see here. So when the infection is at your lymph nodes in this particular region, then it is your lymphadenitis. Then because it is in the nodal areas, it will become later pus filled because of your bacteria or virus, whatever that has been infecting. And then there will be obviously swelling, enlargement, and it is also painful. And then there is lymphangitis. So, an inflammation in your lymph vessels. If throughout your vessels, or any particular part of vessel in a particular tissue in a particular region also is possible. So, that is your lymphangitis. This is mostly by bacterial infection on the skin. It can also be caused by viral or fungal infections. So, then we have lymphatic filariasis, a parasitic infection that can cause lymphedema or tissue swelling due to lymph, or elephantiasis. This is skin thickening and swelling and accumulation of your lymph. So this is the parasitic infection. So not only because of bacteria, virus, or fungi, even parasites can cause infection in your lymphatic system. So this is either lymphatic filariasis, or it can also be called as elephantiasis, because mostly this occurs in your limbs, especially in your legs. So when such infection has been continuing in the body, then the swelling can become so much that your legs or your limbs can swell up to 2 to 3 times of even 4 times of your normal leg size. So then the leg will resemble the leg of that of an elephant. It can become that huge. So it is called as elephantiasis. Lymphoma, a cancer of the lymph nodes that occurs when lymphocytes grow and multiply uncontrollably. There are several different types of lymphoma. Lymphoma is the cancer of lymph nodes. So, in your lymph nodes, you also have your lymphocytes, your immune cells, etc. So, so every cell, so because these are lymphocytes and immune cells, sometimes their regulation to either proliferate when there is an infection or something, few cells need to be proliferated in order to control the infection, or attack the pathogenic organisms. So if this system or this regulatory mechanism is somehow not functioning properly in a balanced way, then it can lead to uncontrolled growth or proliferation of your lymphocytes, and this is called as cancer of your lymph nodes. That is nothing but lymphoma. So your basic symptoms of any kind of infection or disease to your lymphatic system is swollen lymph nodes, painful lymph nodes, redness or red streaking of your skin over your lymph nodes, then fluid that drains from the nodes into the skin. So the fluid now, because it is so much, or if there is any obstruction, if there is no normal flow of your fluid, it will now start draining into your skin, and then your edema will be formed. So then tissue swelling, skin thickening, scrotal swelling, etc. So all these are your symptoms of your lymphatic system infection or disease. What are the treatments? So your basic treatment would be antibiotics depending upon the type of infection, whether it is bacterial, viral or fungal. So you will be given antibiotics or antiviral, antifungal drugs. Then painkillers to control the pain, anti-inflammatory medicines to reduce the inflammation. So because of the uh, the pus filled lymph nodes, or because of your swollen lymph nodes and your pathogens that are growing in that area, there will be inflammation. So the redness or your red streaking of your skin over the lymph nodes are a symptom of your inflammation, so that has to be brought down. So there will be anti-inflammatory medicines that will be prescribed. Then cool compressions to reduce inflammation and pain. Then compressions to promote blood flow and encourage healing. So your hot and cool compresses both apply here. So uh, due to the excessive swelling of your lymph nodes or drainage of the lymph fluid into your skin and other tissues, this might obstruct your blood flow, or your blood vessels get constricted due to the swelling. So then your blood flow to that specific region or that specific tissue also will get affected. So at that time your tissues will lack oxygen supply and other uh, cells and proteins of your blood. So then your tissues will get damaged. So that is also a very common thing in the chronic condition. So for home remedies for this is all hot and cold compressions. So this is about the lymphatic system and lymphatic system infections. Next is mucosal surfaces. Mucosal surfaces are considered crucial to the immune system because they act as the primary interface between the body and the external environment. So we have several openings and entrances in the body: your nasal opening, your oral cavity opening, your reproductive system opening, your excretory system opening, etc. So all these openings are easily prone to infections and easily prone to any kind of foreign element or any kind of pathogen or microorganisms that can easily enter through these entrances. So most of these entrances are covered with mucosal layer. So these are the mucosal surfaces. These help in the immunity, in giving you immunity against several pathogens that you can be exposed to. So this is basically an interface, or the first layer of barrier between your body and the external environment. This provides a specialized barrier, excuse me. This provides a specialized barrier that actively defends against pathogen through a combination of physical barriers like mucus and a complex array of immune cells, making them a vital first line of defense against the infection. So it's not only the mucus that is present here. There are several cells involved in it, and few of your immune system cells are also involved in this surface layers, because uh, only mucus or anything secreted in the mucus is not enough to be effective protective barrier. If by chance, even through this, if there is entrance, or if the pathogens are trapped in your mucus, then you need your immune system cells to act upon them or clear them or attack them. So it comprises of both mucus and immune cells. So what are the functions? It's a barrier function. Mucus produced by the mucosal surfaces traps and eliminates foreign particles, including bacteria, virus, preventing them from entering the body. So it is not only for your bacteria, virus, or microorganisms, anything that can be foreign to your body, even dust particles, to a certain amount of, to a certain extent, your smoke or your, anything that is uh, that you're exposed to will get trapped in your mucus layer. So it will not uh, your body will not get exposed to it immediately at a higher extent. So along with this, obviously it is a protective layer against your bacteria and viruses. Then immune cell population in your mucosal surfaces. Mucosal surfaces are densely populated with immune cells like lymphocytes, macrophages, and dendritic cells. So there are different types of your immune cells, your lymphocytes, your macrophages, your dendritic cells, your neutrophils, your NK cells, mast cells, etc., which we might have discussed in the first week. So out of these, most importantly, lymphocytes, macrophages, dendritic cells are present in most of your mucosal surfaces. These are specifically adapted to detect and respond to the pathogens at these sites. So at specific locations, there are specific detection mechanisms, patterns that are associated in identifying these pathogens or antigens or any foreign substance. So these are all detected by your cells, your lymphocytes, and then your macrophages and dendritic cells, and then they are cleared, or appropriate signaling will be started. So here you can see this is your lumen. So both your anything that is exposed, even your intestine, your gut, the endothelial layer is the most innermost layer. So that is also surrounded with the mucus layer, because once you inhale or eat or inject anything, it passes throughout the length of your tube. So that is the reason throughout it is present, your mucosal lining. So this is your lumen. These are your endothelial cells. This is your epithelial barrier, and this is your lamina propria. So here you can see just below your endothelial cells and your epithelial barrier, you have your immune cells that are present. Your mucus layer will be here. So if anything that you're being exposed to, any type of toxic substances or any virus or bacteria, this will be acted upon by your macrophages, dendritic cells, and neutrophils by appropriately identifying the antigens of them. So this is the function of your most important function of your mucosal surfaces. So you can see in this picture, your mucous membrane is your mucosa, and the various types of mucosa are your nasal mucosa, that is through your nose, your nasal layer, and then your oral mucosa through your mouth. So entire oral uh, tube and oral cavity is all with your mucus lining. And then your esophageal mucosa, as I said, it continues throughout the length of your opening. So then the mucosal lining is also present throughout the length. So at different regions, based upon the organ it is lining, it is named according to the organ. Otherwise it's more or less the same mucosal layer. So your nasal mucosa, your oral mucosa, esophageal mucosa, bronchial mucosa for your lungs, and then gastric mucosa in your stomach, and then your mucus layer in your intestines. So this is all throughout your anything that can be exposed via your inhalation and your eating. Next is there is another mucosal tissue actually. So this is your mucosa associated lymphoid tissue, also termed as MALT. So under MALT again you have specific tissues, lymphoid tissues. So specialized lymphoid tissues located within the mucosal linings, like the gut associated lymphoid tissue is called as GALT, GALT, and the bronchus associated lymphoid tissue is your BALT, plays a key role in initiating immune responses against antigens encountered at mucosal surfaces. So as you we saw earlier, this entire thing, all your cells associated with your mucosal layer will come under your mucosa associated lymphoid tissue. So in your under this you again have, depending on the region it is present, you have your nasal associated lymphoid tissue, your NOLT, and then for your tonsils, adenoids, salivary glands, cervical lymphoid nodes, and then this is nasal or intranasal, and then your BALT, which is for your lungs, that is your bronchoalveolar, and for the lymph nodes present in that region. Then your GALT, that is your mesenteric lymph nodes, lymph follicles, your Peyer's patches of your intestines and stomach lining, and then genital tract associated lymphoid tissue, inguinal lymph nodes. So you can see at every specific organ you have all these lymphoid associated tissues, your intranasal, pulmonary, oral, sublingual, intragastric, your rectal and vaginal. All your openings of your body is associated with your mucosal layer. This is your first line of barrier and defense. If this is not present, you will be very easily subjected to infections and exposed to various types of pathogens. Then IgA production. So the primary immunoglobulin produced at your mucosal surfaces is your IgA type. There are several different types of immunoglobulins, IgA, G. So all out of all these, IgA is for your mucosal surfaces. It helps to neutralize the pathogens by binding to them in the mucosal secretions. So this is effective, and this is also helpful in several of your pathogenic uh, antigens, not only your pathogens but also neutralization of your any of your toxic particle that has been uh, acquired or taken up by your mucus layer. Then commensal microbiome interaction. So your mucosal surface is uh, not only comprises of the mucus and your cells, your own body cells, that is your lymphocytes, macrophages and all. It also comprises of a residential microbiome, that is your microorganisms in your mucus layer. So we are well aware that we have a lot of microorganisms that we host, that is either in your oral cavity, buccal cavity, or in your intestines, your probiotic bacteria, etc. So in the same way, in your mucus also there are residential microorganisms that help your immune system and help you tackle uh, the invasion of your pathogenic microorganisms. So these microorganisms do not cause any disease unless there is any alteration in in your body's immune system regulation. They are helpful. It's helpful for both the host and the residential microbiome. So these are beneficial bacteria. Mucosal surfaces are also home to a diverse community of beneficial bacteria. These are called as commensal microbiome, which can influence immune responses and promote overall health. So there are the main ways how these bacteria help us is, first is it can help regulating your immune system, uh, helping you recognize certain pathogens or recognize certain antigens in the pathogenic bacteria, and it can um, it can inhibit the quarant sensing mechanism of your pathogenic microorganisms. So your quorum sensing is nothing but uh, signaling or communication between your microorganisms, especially bacteria. So when some pathogenic or infectious bacteria, when they communicate with each other by secretion of certain chemicals or certain secretogens, they can establish an infection. So this will also be inhibited by your residential commensal microbiome. Then colonization resistance. So because your mucosal layer already contains a specific amount of microorganisms, there is not much space or not enough, area for your pathogenic bacteria to easily enter your mucosal layer or easily attach to the mucus layer and start its colonization. So in that way also it's like a competitive uh, inhibition for space and even source. So not only this, not only in immune system point of way, other than this, it also helps in digestion of several dietary compounds such as your short chain fatty acids, amino acids, etc. So these bacteria produce certain chemicals which help in digestion of these, and this nutrient is then absorbed by our body. So it is also helping us to maximum or efficiently absorb the nutrients and energy from our food source. Then it also helps in regulation of your Treg cells differentiation, etc. So basically it helps in regulation and prevention of uh, pathogenic bacteria infecting your body, and regulation of your immune cells. Okay, so this is how your mucosal surfaces help your body's immune system to build up against several pathogenic bacteria, virus, etc., and how they protect your uh, openings, your entrances, your body from different types of invasions, and how they are present at each and every specific area that can be easily exposed to such pathogenic organisms. Next, next is mice irradiation experiment. So mice irradiation is one of the major models for immune cell or immunology research. So the mice is irradiated with UV mostly to deplete all its own mice's own immune system or immune cells, depending upon the you know uh, area of the mice exposed, or the organs or of the region of the mice exposed, or the time and dosage of irradiation. So basically this helps in different ways that you need to when you need to specifically see the effect of particular type of dysregulation, particular type of immunity development, your innate immunity, or your particular type of organs or part of your immune system, if you want to be very specific to check, because all these work in together as a network. All the immune cells, immune systems in your body, your different immune organs, your cells, your um, B cells, T cells, everything work together as a collective to eliminate or to prevent from your diseases or other things. So when you want to see one particular thing how it functions, or you want to manipulate one particular uh, part of your immune system, then you use your rodent irradiation. So the most common application of your rodent irradiation is to destroy the bone marrow and the other hematopoietic progenitors. This is myeloablation, either for immunosuppression or before replacing the immune system with a donor grafts. So as I said, if you want to particularly check like clinical applications of anything that you have developed, if you want to graft from a donor, if you want to inject one particular type of cells or some modified T cells or CD cells, positive cells, if you have manipulated something and then you want to inject it and check its response in the mice. So this is very helpful. So first you destroy the bone marrow and other hematopoietic progenitors, and then this is majorly for your immunosuppression. In immune research, irradiated mice are primarily used to study the immune system by essentially wiping out their existing immune cells. So, as I said, uh, the complete immune cells of the model organism is wiped out. That is, it is made nullified or it is removed by irradiation. So this allows researchers to then transplant new immune cells from donor mice or even humans, thereby creating a model to study specific immune responses. Like I said, specifically your B cell functions, your B cell maturation, or your T cell responses, specific things without the involvement of other lymphatic or immune system. You can study only one particular thing. So then also you can check for transplant rejections, donor graft rejections, and then development of immune cells in a controlled environment. So in physiological conditions, you do not know which signaling is affecting your whatever new inoculation that you have done in the animal or in the model. So you never know if, because you are injecting something from a different species or even a different organism of the same species, it will always generate a immune response. So this graft rejection or donor rejection is all because of the immune response. If you don't want that, if you want to suppress that, and if you want your model to accept your grafts and accept your newly uh, injected cells or tissues or anything of your immune system, so then you have to suppress the immune system or immune response of your host or your model organism. Only then this can effectively work in that model. So that is what is meant by a controlled environment, where you can use whatever specific things you want to use, and the host's physiological immune system will not interfere in this process. So this process is often called as bone marrow reconstitution, as the bone marrow is the primary source of immune cells. So we saw that bone marrow is also part of your lymphatic system. So many of your immune cells which are proliferated or which are again activated, triggered for more production, are from your bone marrow. So if there is even failure of the cells which are already present to attack your uh, foreign material or your pathogen or any new cells that have entered your body that is foreign to the body, your bone marrow can always be triggered and activated to produce more immune cells and several other immune system signaling. So this, that is why your bone marrow is also destroyed for the by the irradiation. So now when you give new cells, new transplant, so your bone marrow reconstitution is done, and then uh, your system is now controlled so that you can see particular effect and you will get your results accordingly. Studying immune cell development. So how this is used in various ways, you can see in the picture. It can be used for B cell maturation and function, antibody responses to test the antibody responses, and then innate immune cell development and function, anything that is related to any part of your immune system, lymph node and secondary lymphoid organ development, and then development of robust HLA restricted T cell responses, your reduce GVHD, increasing community access, that is for this mice, and then development of standardized assays and shared controls. Engraftment rate can be checked, and then renewable sources of human cells and tissues for grafts. So all these can be done using this mice. So studying your immune cell development by irradiating the mice and then transplanting the donor bone marrow. The researchers can investigate how immune cell populations develop and mature under controlled conditions. Exactly whatever we just discussed. And then investigating graft versus host diseases. So, in several treatment conditions, we have organ transplant or even tissue transplant or grafts that we use as treatment methods. So, before that, we have to, check or make ensure that it does not undergo graft rejection or it does not generate lethal immune response in the host. So, when transplanting immune cells from a donor mouse, with a different genetic background into an irradiated recipient. Researchers can study the development of GVHD, that is graft versus host disease, a serious complication that can occur after bone marrow transplants in human. So, as I said, even if the organism belongs to the same species, each person's or each organism's genetic makeup is different. So, the slightest of the genetic difference can be enough to create an immune response, generate or activate an immune response in the host host model or host organism, even in humans, so that this immune response will now eliminate that externally added tissue or organ or anything or graft. So this has to be prevented, because in several of the transplanted patients, they can even it can even lead to the death of the patient because of graft rejection, because of highly activated immune system. So these kind of graft versus host diseases can also be checked by transplanting the immune cells of the donor to the irradiated mice. Then modeling autoimmune diseases. So the autoimmune diseases are when the body's own immune cells attacks self cells or self tissues. So always there is, when the immune cells are matured or proliferated or activated, there is always elimination or selection, so that your body can recognize your cells but it should not attack your own cells. So that is how your immune system is developed. So but sometimes due to some conditions or some errors in the genetic makeup or some mutations, the these cells fail to recognize or fail to differentiate between self and non-self cells. So then they start attacking your own cells, and your immune system is now your own body's enemy, so it starts killing your own cells. So then it develops into autoimmune diseases. So, by selectively manipulating the immune system of the irradiated mice with donor cells, researchers can create models for studying autoimmune diseases where the body's immune system attacks its own tissues. So, because they have irradiated the mice, now the immune system will not attack self or non-self. So now they can, specifically make some changes or do some genetic remodeling, and then inject the donor cells to check whether this autoimmune response is being suppressed or not. Then cancer research. Irradiated mice are used to study tumor growth and immune responses to cancer by transplanting tumor cells into mice and observing how their manipulated immune system interacts with the tumor. So, uh, many of the treatment strategies today are involved, other than your chemo and radiotherapy for your cancer and your tumors, is you're enhancing your own immune system. It is by either enhancing or your regulation of your immune system, or by modifying your uh, T cells or your B cells, anything, your antibody treatment. So all this are manipulation of your immune system to eliminate your tumor growth or your cancer. So this research can also be easily done in irradiated mice, because now you have your manipulated immune cells or manipulated immune system that you want to check whether it is more effective than your normal immune system on the tumor. So this kind of research can also be done in the irradiated mice. Next is testing immunotherapies by using irradiated mice with transplanted human immune cells, that is humanized mice. So you can, because now the immune system is completely collapsed in these irradiated mice, it will not recognize any cell as foreign. It does not have the capacity to recognize and attack foreign cells. So you can even transplant human immune cells, and the host will accept it, and you can see how it functions in the model organism. So that is what is your transplanted human immune cells. This is also called as humanized mice. So researchers can test the efficacy of new drugs and therapies targeting the immune system. So all these are at the end stages where you have developed some drugs, developed new targeted therapies for your either your immune system, your autoimmune, cancer research, anything. So your end result, you want to check how your immune system is acting upon this, or how your immune system reacts to these kind of things. So your irradiated mice is really useful for checking the efficacy of 2 3 drugs. If you have made as candidates, you can see which one is more effective. All these things can be done. So the major things that you have to remember while performing such irradiated mice experiments are dosage and lethality. So the dose of radiation used to, wipe out the immune system of your mice is very important. So this has to be given with sublethal doses, partially suppressing the immune system, while lethal doses completely ablate the immune system, requiring donor cell transplantation for survival. So when you do partial, like I said, based on the region you are irradiating, based on the time you're irradiating, based on dosage you're irradiating, the immune system level can be suppressed to a particular level. So if you need partial suppression, you will always go with sublethal. Otherwise, if your uh, if your host or your model organism has zero immune response, then whatever you do, it might either be because of your experiments, or it might be due to a natural uh, infection or pathogen entry to the model organism, and it will easily die. So when you're giving most of the time sublethal doses to partially suppress the immune system, can be done. If there is a need for a complete depletion of the all the cells and all the progenitors, bone marrow, everything of the immune system, so that becomes lethal. So that time you have to give a transplant of the bone marrow or any other cell transplantation for the model organism to survive. Otherwise it will not live for any experimentation or further research. So you should know whether your experiments require a lethal dose or sublethal dose, partial suppression or complete suppression. If it is specific suppression of only one type of cells, or only bone marrow, or only lymph, something like that, then you have to check in if it is whole model or particular region, so that all these different irradiations can be done. So you have to be very particular in knowing what kind is actually required. If there is no requirement of complete suppression, then always go with sublethal doses. Then mouse strain selection. Different mouse strains have varying sensitivities to radiation. So every mouse strain is different from each other. Their genetic makeup is different. Their tolerance is different. Their survivability is different. Uh, their endurance is different. So based on your experiment, the need of your experiment, how different strains react to the type of experiments you give, you should know, and the endurance of it you should know. So based on all this, you have to pick a very appropriate strain. And particularly for radiation, few strains are more tolerant, few strains are very sensitive. So the doses also should be standardized according to the strain you have chosen. So different strains have different sensitivities to the radiation that needs to be considered when designing the experiments. And then of course there are ethical consideration. As with any animal research, using irradiated mice always raises ethical concerns regarding animal welfare. So researchers must adhere to strict guidelines and minimize suffering. So the uh, use of animal models is always under ethical guidance. So you cannot uh, randomly go and start experimenting on any kind of living organism. So you need your ethical committee approval, you need your grants commission approval to do these experiments, and you need to give them proof or show them proof that this experiment is required for this project. If there is no requirement, you cannot do unnecessary experiments using living organisms. So everything has to be properly done according to the ethical committee board. So even your irradiation experiments, anything that you are using a living organism will come under your animal welfare. So you have to follow your guidelines, and even though we have to sacrifice these animals for purpose of research and science, you should always see that the animals don't suffer. So even the method to sacrifice it is also done in such a way that it can um, it can take a minimum amount of pain, as minimum as possible. So all this have to be considered. Next is thymic selection. Thymic selection is a process that occurs in the thymus to differentiate the immature T cells into mature T cells. This process involves positive and negative selection. So your T cells, there are naive T cells and then they mature. So during this process, the T cells have to be selected. Which T cells are eligible to mature, which T cells are not eligible, then they have to be discarded from the system. So this process of selection is called as thymic selection, and it involves both a positive selection and a negative selection. So this will ensure that the T cells can respond to foreign antigens while also being tolerant to the body's own cells. So like we discussed about the autoimmune disorders or diseases, the the immune cells must always be able to differentiate between self and non-self. That is, your own body cells and your own body antigens or anything, and something that is not belonging in your body naturally. Even the cells from a different person or anything, an animal or uh, an insect, anything, if it is taken up or by mistake, if it has entered your body, then that has to be detected as non-self. Only then it will be discarded, only then that if it has any negative effects, that will be eliminated. Otherwise, anything that is foreign to your body has probability of causing harm to you. So your immune cells have to differentiate between self and non-self, and once it differentiates this, it should try to eliminate the non-self antigens or non-self cells. So that is why both positive and negative selection is required. So positive selection ensures that T cells can respond to foreign antigens. That is, it can recognize that something is not from your body, something is foreign. It eliminates T cells. Positive selection eliminates the T cells with TCRs, that is T cell receptors, that are too dissimilar from self MHC. So anything that is very dissimilar from yourself, your positive selection will try to eliminate that, the TCRs. And then it also promotes the selection of naive T cells for maturation. It involves weak interactions between TCR and self peptide MHCs. So, if your cells have to recognize your own cells as self cells, then it has to, bind, and it has to interact somehow with it. So, you can see here in this, you can see that this is your own cell, this is your thymic cell. So this is your T cell. Your T cell receptors are present, and it is interacting with the MHC of your thymic cells. Your thymic cells here are nothing but your self cells. So it has some sort of interaction, but this is a weak interaction. There is moderate binding. So this will undergo positive selection, and this T cell will survive. Now coming to negative selection. Negative selection enforces self tolerance. Negative selection eliminates T cells with T cell receptors that are too similar to self peptide MHCs. So here you can see the difference. Here it eliminates the T cells which are too dissimilar to your self MHC, and here it eliminates that are too similar to your self MHC, because this is for self tolerance, and then causes negative selection by apoptosis. So if there is strong binding and like it's too similar to your peptide MHCs, and there is it recognizes your own self cells very effectively with stronger binding, so that further immune cell response might be triggered, then this cell will be harmful to your body. So that's why this cell should be now eliminated by negative selection. So here it involves strong interaction between TCRs and self peptide MHC complexes. So here you can see, here also your T cell is now binding to your thymic cell through your MHC molecules. So now this binding or interaction is very strong, and this can trigger an immune response. So such kind of T cells which can recognize your own cells to trigger an immune response is not required, because if it is there, then your body can't tolerate self cells. So that is enforcing self tolerance. So this has to recognize it, but it should not be so strong enough to trigger an immune response. Then only it can survive. That is your positive selection. But if it is too strong to trigger an immune response, then it will be selected for cell death. That is nothing but apoptosis. So normally also your cells, when there is antigen, now this is your cells that is not self, that is any cell that is not of your own body, either a cell that has been infected by a virus, or either a cell that has been mutated, like a tumor cell, or something that is abnormal from normal physiological condition. So that will now be selected by your mature T cell, and that will uh, help in recognizing this cell as non-self and foreign and antigenic, and now this cell will be uh, discarded or eliminated, or it will be killed. So this is what is required. So for this kind of both positive and negative selection is very important. So that is your thymic selection. The process that ensures only non-self reactive T cells surviving during development in thymus is called as negative selection. So as we said here, it enforces self-tolerance. That means it ensures that non-self reactive T cells surviving during development in the thymus. Okay, so that is your negative selection. During T cell maturation in the thymus, developing T cells encounter self antigens by thymic epithelial cells. If a T cell, your self antigens are present on your self cells, that is cells from your own body. If a T cell strongly interacts with a self antigen, that is nothing but it is reacting with the self antigen, so it is self reactive. It triggers a programmed cell death pathway, eliminating that T cell, because it should not uh, interact so strongly with a self cell or a self antigen or a self MHC. So if it is very strongly interacting, it has to be immediately eliminated, otherwise it will cause self damage. So this kind of T cell will now be eliminated. The entire T cell repertoire, that is that entire set of T cell which does a similar kind of strong interaction, the entire set will be now eliminated. Thus, it prevents autoimmune reactions. So here you can see number of thymocytes that usually develop and survive are, most of them, the death can be by three ways, that is death by neglect, or due to positive selection, or due to negative selection. In the positive selection, you should also remember that the cells which does not recognize your own cells as self cells, even those cells has to be discarded, because every immune cell in your body has to recognize your self cells. If it doesn't have the capacity, or if it doesn't have the T cell receptors to recognize your self cells, even that can be harmful. So both in your positive and negative selections, your TCR thymocytes, TCR will be sorry, your T cells will be selected. So here you can see the selection, both positive and negative occurs at different regions. Your positive selection occurs at cortex and your negative selection occurs at your medullary region. So your negative selection occurs primarily in the medulla of the thymus. The mechanism is self-reactive T cells undergo apoptosis when they strongly bind to self-antigens that is presented by the antigen presenting cells. Result, only T cells with receptors that do not strongly recognize self-antigens mature and leave the thymus. So this is how your mature T cells are now eligible enough to go to your travel throughout your body and detect and protect your body. So here you can see in your cortex, your positive selection is occurring. Your cells which can weakly interact with your cells are taken and sent for next step of maturation. And now in this step, if your T cells are binding strongly with your self cells that are presented with your antigen presenting cells, those will be killed, and ones that do not bind strongly will be leaving your thymus after maturation. Okay, so this is how your positive and negative selection play a role in uh, protecting your body against the external pathogens, and also protecting your body by preventing self damage. Next are M cells. M cells are specialized mucosal epithelial cells that lack cilia, do not express the polymeric Ig receptor, and appear flattened with surface microfolds on the luminal surface. So most of your lining cells will have cilia, which helps in increasing your surface area, etc. But your M cells are present between these epithelial cells, and these are also your epithelial cells, but instead of cilia, they have microfolds. So they are also called as microfold cells. M cells, also known as microfold cells, play a crucial role in the immune system by acting as gatekeepers that actively sample and transport the antigens from the intestinal lumen to the underlying lymphoid tissue. So intestinal lumen, like we saw here, even your intestinal lumen are also lined with cells. So here you'll have in your intestine, you will have ciliated cells, and in between you will have one or two M cells. So, like how here you have your lymphoid tissue system present immediately, in the same way, there also you'll have your lymphoid tissue system. So, these cells help in processing the antigens and uh, anything that is foreign, and send it to the underlying lymphoid tissue where your uh, immune cells will now act on it, essentially initiating immune responses by delivering potential pathogens or foreign substances to the immune cell for recognition and defense. So it mainly acts as a gatekeeper to let in or not let in, or if it is in your intestines, it has to be processed, so it will now allow your pathogens or potentially pathogenic particles to go to your lymphatic system. It is present primarily in the epithelial lining of the Peyer's patches, and which are specialized lymphoid tissues in the small intestine. So the function is M cells actively take up antigen from the gut lumen, that is the intestinal lumen, through the through their unique microvilli structure. So your microfolds help in taking up the antigens and delivering it to your lymphoid tissue underlying it. Then transport them across the epithelial barrier to the underlying immune cells like macrophages and dendritic cells. Immune reaction initiation. By delivering antigens to the immune cells, M cells trigger the development of specific immune responses against ingested pathogens or food allergens. So, basically, they are initiating an immune response to the foreign substances by making these antigens and foreign particles available to your immune cells to recognize them and trigger an response. Potential vulnerabilities, while vital for immune surveillance, M cells can also be exploited by pathogens as an entry point into the body due to their antigen sampling function. So, as they are called gatekeepers, it is also an entry point. Even though it is useful for us in a way that it helps in delivering it to the immune cells, at the same point it is also an advantage for the pathogen because it is able to now penetrate into your epithelial lining. So either way, depending on the type of the antigen or type of the pathogen, how it attacks, what type of cell it attacks, it can be exploited by some pathogen. So mostly this is how it is present. These are your ciliated cells and these are your microfolds M cells. So you can see it can ingest or it can deliver the antigens, pathogens to your lymphoid. Your lymphoid tissue cells will be here. These are your dendritic cells and this is your macrophage. So these are ingested by your cell by a process called transcytosis. So transcytosis is it will take up from one end and it will deliver the materials, the cargo to the other end. So now once sent to the dendritic cells, now the dendritic cells will process these and it will induce an immune response. So usually your dendritic cells also have a periscoping property that it can extend its appendages between your epithelial cells to pull these inside, but you can see it's not so easy. So your through your M cells, this is very effective. It enhances the efficiency of the periscopic nature also. So the luminal capture through M cell transcellular pores. So this M cells has the property of forming pores, transcellular pores, which help your appendages of the dendritic cells to easily access these antigen. Then translocation of small soluble antigen via goblet cell associated antigen passages. So through your goblet cells also which are present, these can be exposed to your dendritic cells, but these will only do it with a soluble antigen. Anything that is soluble, proteins or hormones or any secretory objects or any um, processed object or anything which is soluble, that can be also sent via the goblet cells. Then translocation through to basolateral sides of the enterocytes. So these are your enterocytes. So translocation can also be done by your enterocytes itself, but this can again be done by done for specific type of antigens or pathogens. So this is common for all types. Anything can pass through this. So now this will also be uh, attacked and recognized by your macrophages, which are phagocytic in nature, and then it leads to a process of phagocytosis and then degradation of all your microorganisms and your antigens or foreign particles. So this uh, your M cells and your epithelial your enterocyte cells are for both microorganisms and particulate antigens depending on the type. Your M cells can almost send anything inside, like any particle or microorganism of any size which is smaller than your cell, it will be able to deliver. But there are limitations for your goblet cells and your enterocytes. Only a specific dimension or specific site of particle it can send for processing by your macrophages. So anything soluble is less than your 10 kilodalton size that can pass through your goblet cell. Only that is possible. Others will not be able to be delivered. So this is how your M cells process, or M cells deliver your antigens, microorganisms, etc. to your lymphoid system for immune cell initiation. Next, antigen phagocytosis. So we know what is phagocytosis. It is a process to eliminate or you know ingest and degrade something so that it does not show its negative effects. So your antigen phagocytosis is your degradation and processing of your antigen, and this is also done in the skin. So phagocytosis in the skin is a process where cells in the skin take in antigens and process them. Phagocytosis is a complex process that plays a role in many processes, including, as in it is involved and it is significant and it is important in many cellular events or many bodily functions, like for inflammation, for tissue remodeling and repair, innate and acquired immunity, extra and intracellular infection. In all these situations, in all these processes, phagocytosis plays a very important role, and it is definitely very important for body's immune response. So what are the cells that are particularly involved in antigen phagocytosis of the skin? In the skin are your dendritic cells. They are the major cells those are involved. Other than this, melanocytes are also capable, sometimes equipped with phagocytic machinery. But your major cells that are involved are your dendritic cells. These cells are professional APCs, that is, they are professional antigen presenting cells. So they process your antigen and make the antigen available or expose to your macrophage or your any type of phagocytic cell to attack it and digest and degrade it. So they take in antigens and link innate and adaptive immune response. Melanocytes, these cells are also equipped with the phagocytic machinery and they can process antigens to a certain extent. Then the basic mechanism behind this is a pathogen is phagocytosed by a dendritic cell. The dendritic cell produces pro-inflammatory mediators to now recruit innate cell immune response. So your DC cell is the first to take up your antigen or your pathogen, and it will ingest it, process it, expose the antigenic region to the other cells, and now these cells will recognize it and start producing pro-inflammatory cytokines. So this cytokines will now enhance the activation or proliferation of other immune cells, whichever is required depending upon the type of pathogen and antigen, and now they will all be recruited in that place to attack the process the antigen or pathogen. Then the DC, the dendritic cell migrates to a lymph node where it encounters T cells. The T cell probes the DC cell to see if it is presenting an antigen it recognizes. If the T cell recognizes the antigen, it becomes activated and then searches for infection. So, as I said, basically, now this is basically like an informer. So it will take this, it has caught something the cell thinks that it is toxic or harmful to the body. So now it has to go and give it to somebody who can take care of the situation, or which it who can recognize and confirm that yes, this is antigenic, this is pathogenic. So that is your T cell, because your T cell has your receptors to uh, bind and recognize them, self and non-self, and pathogenic, antigenic, etc. So once your T cell now recognizes it as antigenic, it has to now start the process to produce more cells and activate T cell response or B cell response depending on the type of response it requires, to now first search where the infection is. That is why the cytokines are produced. Uh, it is like it is an attractant for your immune cells to now see that this is the site of infection, because pro-inflammatory cytokines are being produced at this region, so they will come to that region and then uh, stop the infection by attacking the pathogens. So this is your basic mechanism. So your dendritic cells in the skin are a type of immune cells that present antigen, your antigen presenting cells, and regulate the immune response. They are found in the epidermis and the dermis and are important in the body's defense against pathogens. There are different types of dendritic cells. The major type are the Langerhans that are found in the epidermis, and then we have the dermal myeloid dendritic cells that are found in the dermis layer, and then we have dermal plasmacytoid dendritic cells also found in the dermis, and inflammatory dendritic cells, a type of myeloid dendritic cells that appears in the inflamed skin and in diseases or conditions like psoriasis, because there is already some infection or disease that is there, and there is a inflammation over there, so they are inflammatory dendritic cells. Melanocytes are nothing but the cells that produce melanin, which is a pigment, and the pigment gives color to your skin, your eyes, your hair, and other parts of the body. So they are found in all these regions, and also in your ears and heart and central nervous system. So these are the major dendritic cells. Now, next is a knockout and a conditional knockout. A knockout refers to a genetic manipulation where a gene is completely deleted in all cells of an organism. So, any particular gene that you want to knock out, or you want to silence, or you want to um, stop it from functioning, and if this is done in the entire system of the model or in all the cells of the model, it is a complete knockout, or in general a knockout. And there is also another type of knockout, which is a conditional knockout. So in conditional knockout, this allows us to delete a gene of interest in a specific tissue, or to delete the gene of interest for a specific time during the development or during particular conditions, whenever you want uh, any nutritional condition or temperature condition. Only in that particular condition the gene will be silenced. It provides more control over where and when the gene is inactivated. And essentially, a knockout is a global gene deletion, global on the whole, whereas a conditional knockout is a targeted tissue specific deletion. So, this is the major difference between a normal, general, complete knockout and a conditional knockout. Complete knockout is global gene deletion everywhere and all the time. Once deleted, it's deleted. And then the conditional knockout is, it is targeted to specific type of cell or tissue of interest, and you can also manipulate or modify or change the time of deletion. If only in a particular condition you want it to be deleted, or in a particular time you want it to be deleted, etc. So the major differences are specificity. A knockout affects the gene in every cell of the organism, while a conditional knockout allows for a targeted gene deletion in specific tissues or cell types. Developmental control. In a traditional knockout, the gene is deleted from the very beginning of the embryonic development. That is, before it can completely develop into an organism, during the embryonic stages itself, the gene knockout is done, and now the entire organism, the gene will not be expressed. Whereas in a conditional knockout, it can be induced later in the development to study gene function in adult organisms. So in conditional, it's not necessary to knock out in embryonic stages unless you want to see the effect during the embryonic developmental stages. You can do it at that time. Otherwise, if you want to see the effects in adults only, then you can also knock it out in the developed organism when it is in its adult stage. Applications are conditional knockouts are particularly useful when studying genes that are essential for embryonic development, as deleting them in all cells could be lethal, while a conditional knockout allows for studying genes function in specific tissues later in life. So, why conditional knockout is important is sometimes you may be studying some genes which, when deleted or which, when knocked out, might not affect the overall growth or survivability of the organism. It will just hinder one specific function, or it will hinder, or it will just give a kind of, deficiency or some disadvantage to the organism. But few genes which are really essential to the body, which you, if disrupted, or if it is non-functional, then it will lead to death of the model organism. So such genes are known as such genes are known as very useful, or like, they cannot be replaced. The function of that gene cannot be replaced or cannot be uh, altered in any way so that your uh, model organism can survive. So such kind of genes cannot be knocked out completely, because if you knock it out, then your body of your organism will not be able to, tolerate this lack of the functioning of that gene. So it will die. So now what you have to do is either knock it out at a particular time, or and then bring it back, or knock it out at a particular condition. So this can be done so that long term deletion is not happening, so that even if it is knocked down now, later that can be again uh, activated, so that your model organism will not be dying. So that is helpful in studying lethal genes which are non replaceable for surviving. So, example, knockout. If a researcher wants to study the effects of deleting a gene involved in cell growth, they might create a knockout mouse where the gene is completely removed in every cell, potentially affecting the entire organism's development. In conditional knockout, if the researcher wants to study the effects of the same growth related gene, specifically in the liver, they would create a conditional knockout mouse where the gene is only deleted in the liver cells, allowing them to observe the effects solely within that tissue. So, not only lethality, also if you want to see the effect of gene deletion only in one particular type of cells, sometimes the effect of the same gene in one cell may not be um, lethal when deleted, and in other cell it might be lethal when deleted to that organ. So it can also lead to organ failure. So if you want to delete certain gene which is only which is not lethal when deleted for a particular organ or a particular type of tissue, or even a particular type of cells, then you can also do conditional knockout. So here again, in conditional knockout, there are two types mainly. So your main knockout or your complete knockout will be done in your germline or your embryonic stage. So you can see the entire model organism. There is no expression of the gene in any part or any cell, and this is throughout the life of that organism, it will be the same. But in case of conditional knockout mice, you can see that particular target cells or particular target organ or tissue where you want the deletion, it is done only in those, and it can be done both in germline or later stages of development. It will be the same. And in case of conditional knockout, there is again non-inducible and inducible. So non-inducible is once deleted, it will always be deleted during the entire growth or development of the organism. But in case of inducible, you can manipulate the time, or you can manipulate at which stage you want the deletion. So it won't be deleted from the beginning or during the entire life of the organism, only at a particular stage of development or at a particular time when you want to do the experiment, you can induce the deletion of that gene. So there are several ways and methods and tools which can be used to do these kinds of knockouts. Next is the stem cells. Stem cells are cells that can develop into many different cell types, including brain cells and muscle cells. They can also repair damaged tissues. So, stem cells have the capacity to develop into different types of cells and differentiate into different cell types. So, like even the major of your vital organ cells, like your brain cells, heart cells, muscle cells, any type of cells, and they can also repair a damaged tissue or injured organ. So there is also stem cell therapy for different organ damage. So there are several types of stem cells, including embryonic stem cells, induced pluripotent stem cells, mesenchymal stem cells, adult stem cells, multipotent stem cells, and hematopoietic stem cells. One second, let me just connect the charger. Yeah. So coming to the types of stem cells, embryonic stem cells come from human embryos that are 3 to 5 days old. So, at this stage, the embryonic stem cells can be harvested and harvested during in vitro fertilization. Pluripotent stem cells that can differentiate into almost any or almost all cell types. Induced pluripotent stem cells. So these embryonic stem cells are normal physiologically present and it can be extracted or harvested. In vitro in the lab, when you generate a stem cell or a pluripotent stem cell, they are called as iPSCs, that is induced pluripotent stem cells. So these are created in the laboratory by reprogramming a normal adult cell. Often these are skin cells. So these are also pluripotent stem cells that can differentiate into almost all cells. So this was done uh, where researchers or scientists have taken an adult differentiated stem cells, adult differentiated cells, that is your skin cells. Now it has already acquired a function, it already has a role. Your skin cell, normally your skin cell cannot uh, you know differentiate into a different type of cells. They can proliferate to form again another layer of skin cell, but these are now reprogrammed. The genetic manipulation is done so that it can acquire a pluripotency. So this is induced pluripotent stem cell. So you can see pluripotent stem cells here. It can almost differentiate or it can almost give rise to any type of cells, your bone cells, nerve cells, blood cells, epithelial cells, muscle cells, etc. Then you also have a totipotent stem cell. So this is almost every type of cell it can give, but your totipotent stem cell can give rise to the entire organism. It has the capacity to become a complete organism. Then you have your multipotent stem cell, which has the capacity to develop into again many types of group of cells, like your blood cells, your platelets, RBCs, white blood cells, etc. So one particular group of stem cells, one particular group of cells can be generated from multipotent type of cells. So you have different again groups of multipotent stem cells, which will each be able to give rise to that particular line of uh, cells. That is, like your hematopoietic stem cells can give rise to all your blood cells. Then you have your unipotent stem cell, which can give rise to only one type of cell, which can keep producing only one type of cell. Then you have your oligopotent stem cell, which can have two or more, two or three types, not entire group also, but not a single also. So a few number of cells can be produced by your oligopotent stem cells, like your myeloblasts, which produce your different types of your white blood cells. Next, so your mesenchymal stem cells. These are multipotent stem cells that can differentiate into multiple cells. So they can be derived from variety of tissues, including your bone marrow, skin, and fat. Adult stem cells can be classified as multipotent or unipotent. Progenitor cells are a subclass of adult stem cells that can self-renew and differentiate. So it can also self-renew itself, and then it can also differentiate into its future cell. Then multipotent stem cell, as we discussed, it can differentiate into a specific range of or specific group of cells within a particular lineage. So essential for body's tissue renewal and repair process. So your blood cells need to be renewed, right? So it's for that. Then your hematopoietic stem cell is also multipotent stem cell. So but this is one specific group, so it has only the capacity to produce only your blood cells. So young immature cells that can make up all the blood cells in the body, usually found in the bone marrow. These are your hematopoietic stem cells. So I hope different types of stem cells are clear. There are embryonic stem cells, induced pluripotent stem cell, what is a totipotent stem cell, unipotent stem cell, oligopotent stem cell, and multipotent stem cell. Then your mesenchymal stem cell, adult stem cells, and your hematopoietic stem cells. Then coming to lymph. So your lymph is your major part of your lymphatic system. This is what runs throughout your lymphatic system. So lymph is a clear or white fluid that carries nutrients, white blood cells, and other substances throughout the body. It carries nutrients, white blood cells, and also other substances. It's part of the lymphatic system, which is a network of organs, tissues, and vessels that help the body fight infections and diseases. Like we saw in the first slide, so here all these lymphatic system throughout all these organs and all these vessels and layers, what is the thing that passes, or what is the thing that is running through the entire system, is your lymph, through all your lymphatic system organs, tissues, etc. So what is the function is, it carries nutrients. Lymph carries nutrients like carbohydrates, proteins, and fats. It carries white blood cells like lymphocytes, macrophages. This helps in fighting infection, that is the major immune cells, and collects waste. Not only does it provide nutrients and nourishment, but it also, and also the immune cells, but it also uh, collects the waste once your pathogens are inactivated, or antigens, whatever damaged cells, debris, whatever is present from your body, all this is also collected via your lymph. So your lymphatic system also helps in excretion of all these. Then filters lymph. Lymph nodes filter lymph and remove foreign material. So that is the major function of your lymph node. So it is where the filtration of your lymph occurs. Now because your lymph carries so many things, your nutrients, your white blood cells, your wastes, your lymph nodes help serve as a filtering unit for your lymphatic system to remove foreign material from the lymph. So it majorly like purifies the lymph of the waste in that it is collecting. Then maintains fluid levels. Lymph helps in maintaining fluid balance or fluid levels in your body. Then how the lymph moves is via your lymphatic vessels. So your lymph vessels collect the lymph from the tissues similar to your blood and carry it to the lymph nodes where the filtration occurs. And then lymph nodes clean the lymph and add more lymphocytes. So it's a kind of replenishment also of fresh set of cells, fresh set of immune cells, or if it's replenishing the system. Then lymph nodes collects lymph fluid collects into a large vessel and then drains into the blood vessel near the heart. So finally, it is draining into the blood vessel near the heart. What is lymph made of? Lymph is nothing but majorly plasma, which is the liquid part of the blood. Lymph also contains chyle, which is fluid from the intestine, and it contains proteins and fats. Like we already know, it contains proteins, carbohydrates, and fats. So this is your lymph, which is the fluid that runs through your lymphatic system. I hope it is clear. So I think we have 10 minutes left. So do you want me to explain any topic again, or if you have any doubts, you can ask me now. If not, we can end the session. Any doubts, anyone? Okay, if there are no doubts, I think we can end the session. Thank you for attending, everybody. See you in the next session.